Okay, anyone want to ask about the script? The yeah,
স্যার
জি স্যার।
স্যার আমার কোয়েশ্চেন 5 এ প্রবলেম ছিল, ওইখানে আমার 1 মার্ক গিয়েছে, আমি বুঝিনি কেন।
Question 5, just 1 minute. কই গেল? Okay. এই দাঁড়াও 1 সেকেন্ড, দিনকে দিন আমি টেকনোলজি ইউজ করায় স্লো হয়ে যাচ্ছি। আগে ভালো পারতাম।
ইটস ওকে স্যার, টেক ইউর টাইম।
না, এটা ভালো না, কিন্তু আগে কিন্তু আমি ভালোই পারতাম। হ্যাঁ, ইদানিং একটু মানে টেকনোলজি থেকে দূরে সরে যাচ্ছি মনে হচ্ছে। মানে টেকনোলজি ইউজ করতেছি কিন্তু স্কিল হারায় ফেলতেছি। ওকে, তোমার ইমেল অ্যাড্রেস কি জানি?
স্যার তাজরি হাসান শ্রাবণী
না,
@g.brad।
পুরোটা লাগবে না, পুরোটা
ওইটা
লাগবে না। ওকে, গট ইট। I mean, checking the script direct from the Google Drive is a pain, but we have no other way. And checking the hard copy script is much more, what can I say, comfortable. That is okay. So, okay, that's it. So, Tazri, so you say that you are confused with question number 5. Mm, okay, let me check first. Okay, Tazri, so half of your logic is okay. So, for example, I mean, from while the white blood cell, I mean, you talked about immune system. তুমি যে ইমেল সিস্টেম নিয়ে কথা বলছো, এই লজিক ঠিক আছে। প্রথমে যেটা লিখছো না, সে লাইকছে ওই যে কিছু নিউক্লিক অ্যাসিড, সাম কি জানি তো, so like say the pathogen did not reach the organ or site of the disease. Okay, I mean, it was not the question, right? মানে ধরো যে ওই মানে ডিজিজড অর্গানে পাওয়া গেছে কি জানি, সেটা নিয়ে কিন্তু আমরা আসলে কনসার্ন না। আমরা কনসার্ন কি কোশ্চেনে যে ওই নন ডিজিজড বা হেলদি হোস্টে কেন পাওয়া যাচ্ছে। হুম, কি বলছি বুঝছো?
ইয়েস স্যার।
হেলদি হোস্টে কেন পাওয়া যাচ্ছে, সো এখানে ওই ব্যাপারটা না, ওটা ইয়া না যে সে তার সাইট অফ কি বলা যায়, সাইট নট নট দ্য সাইট অফ এন্ট্রি, তার সাইট অফ ইনফেকশনে মানে আই মিন whether that pathogen leads to the site of infection or not. ঠিক আছে, বুঝতে
ওকে
পারছো?
স্যার। জি স্যার।
তো এই অংশটা ইরিলেভেন্ট আর কি।
এমনি বাকি কিছু ঠিক আছে, জাস্ট ওই পার্টটা জন্য।
Yep.
ওকে স্যার, থ্যাংক ইউ স্যার।
Welcome. The question was very easy, I think. It was very easy.
স্যার
Yeah.
স্যার আমি হচ্ছে ইয়ারটা পাইনি, মার্কস পাইনি স্যার।
তুমি মার্কস পাওনি? তুমি মার্কস কেন পাওনি?
স্যার আমি হচ্ছে অ্যান্সার আপনাকে মেইল করেছিলাম, আমি পিডিএফ করে দিতে পারিনি তখন স্যার।
ইয়াল্লা, দাঁড়াও 1 সেকেন্ড, দাঁড়াও আমার স্ক্রিপ্ট গুলো চেক করি। ওহো, ইয়াপ। আচ্ছা, তোমারটা তাহলে আমি একটু পরে দিয়ে দিব, হ্যাঁ?
ওকে
ওকে, yeah, I have your script, but okay, I have your script, কিন্তু ওই যে মানে ভুলে চেক করতে ভুলে গেছিলাম আর কি। ঠিক আছে,
ওকে স্যার,
yeah,
সমস্যা।
I have your script. ওকে, আর কেউ কিছু বলতে চাও? এনিওয়ান, এনিথিং এলস?
Excuse me, sir.
Yeah, Kevin.
What is a violence factor?
Oh, what is violence factor? Okay, so you can say violence factors are all those particles of a, or all the parts of a pathogen by which that pathogen cause disease. Okay, say for example, say Vibrio cholerae. Okay, just 1 second. Um, okay, say Vibrio cholerae. Okay, Vibrio cholerae. Hmm. So Vibrio cholerae, it cause cholera by what? By toxin, by producing a toxin. So that toxin is a virulence factor of Vibrio cholerae. Okay. Vibrio cholerae also have other virulence factors. So for example, Vibrio cholerae, I mean, there is some protein in the surface of Vibrio cholerae, okay, by which it can attach to the tissue where it infect. So that proteins or that surface material is also a virus factors. So everything of a pathogen by which it cause disease is virulence factors, or are virulence factors. You got my point?
Okay, sir. So does it mean that we can say that the incubation period is also a violence factor?
Now, incubation period is not a virulence factors. Okay, I mean, if the incubation period is not a part of the structure of the pathogen, right? It's just a time. So
Okay,
virulence factors
got
are
any?
the structural part of the. I mean, okay. If we want to say more easily, factors are a structural part of the pathogen. Directly a structural part. Say, for example, okay, you can argue with me that okay, sir, Vibrio cholerae is producing a toxin and releasing it, then the toxin is affecting the tissue. Then it's not a structural part, right? I mean, it is producing the toxin, then it is releasing the toxin outside their cell. So it's not a part no more. Well, the Vibrio cholerae, how Vibrio cholerae produce the toxin? Vibrio cholerae bacteria, they have the gene. You all know what gene is, right? এই জিন কি সবাই জানো তো না? জিনের বাদশার জিন না কিন্তু, হ্যাঁ? কোন জিন জানি? It's not the জিনের বাদশা, it's what? Okay, Kevin, you will not understand what জিনের বাদশা is. It's like a local joke. Okay. Anyway, we all know what is gene, right? So gene is, I mean, like, okay, I'll first answer Kevin's question, then I will describe what actually gene is. Okay, so Vibrio cholerae is producing the toxin. How it can do that? Vibrio cholerae should have that DNA in it, by which it can produce the Vibrio cholerae toxin. So basically, that gene is a structural part of the Vibrio cholerae, right? It's a part of its DNA. Okay. So incubation period is the time required for the pathogen where they like do the damage. Okay. So it's not the virulence factors. Kevin, you understand my point?
Yes, I've got a new, sir. Thank you, sir.
Okay. I mean, if you have any confusion, feel free to ask. Okay. So never hesitate to argue with me. In science, we should argue. We should ask question. We should fight. Okay, this is science. Okay. By the way, I was asking that what gene is. Yeah.
I think it is clearer now. I was confusing the two.
Okay. So you are clear now.
Yeah, I think it is clear now.
Okay. Good. So, for example, the COVID-19. Okay, I'll give you another example. The COVID-19 spike protein. Did you heard about the spike protein of the COVID-19? You guys heard, right? I mean, you saw the photo which circulate in the web. COVID-19 is round and it has some Skype. Sorry, what it's called? Spike. এই কাটা কাটা থাকে না COVID-19 এর বডিতে. The spike is actually, I mean, in the spike, actually spike is a violent factor, not the whole spike. Basically, in spike, okay, if you can say the whole spike. Okay, the spike help the COVID-19. Those are spike, right? Spike are pointy. So spike to katar moton. So by spike, the COVID-19 virus can attach with the host cell. So spike is a violence factor because it helps them to attach with the host cell. Okay, so spike is not the only violence factors of COVID-19, right? It has other violence factors. It has some gene in it by which it can cause the disease further. Okay, usually all the pathogens. Okay, they have several viral factors by which they cause the disease. Okay, so anyway, so okay, guys, you guys all know what gene is. If anyone have any confusion, you can ask me.
Yes, sir. We know
Gene,
what.
জানো না?
Yes.
একটু একটু বলবা জিন কি? Can you just briefly explain? মানে just বললেই হবে যে okay, what you understand by gene? GENE, the word gene. Yeah. Hello.
It's like the Namki, all the characteristics that we inherit from our parents that make us who we are. That's like
Yeah.
a functional unit of that. So all of these make up the so I'm the person that I am right now because of my genes. And it's different from say Apnar genes or any other person in this classroom's genes. So every person's genes is something that makes them the person they are, and it differs from people to people, and we inherit them from our parents during.
Okay. Okay. You are right. Okay. So, but I will add something with it. Okay. So like um, say like okay, I mean your explanation is okay. মানে তোমার এমনি ধারণা ঠিক আছে. আমি just একটু add করতেছি আর কি, হ্যাঁ?
Yeah, sure.
So what is gene? So for example, the cholera toxin. Cholera toxin is a protein. Cholera toxin is a protein. We have lots of protein in our body. Cholera toxin is a protein. Okay, we don't have cholera toxin in our body, but it's a protein. So for example, insulin. Okay, we all know what insulin is. It's a hormone. It's a protein. Insulin is also a protein. So for every protein, there is a specific part in our DNA. Every protein, there is a specific part. Okay, so protein is the product and gene is the, what can you say, gene is the like a information. I mean our DNA is a, I mean, do our DNA produce protein? If we say that it's wrong, right? Our DNA do not produce protein. Our DNA is the information, and following that information the protein is produced. ডিএনএ আমরা যদি বলি ডিএনএ প্রোটিন তৈরি করে, এই কথাটা কিন্তু ভুল। ডিএনএ কি প্রোটিন তৈরি করে? ডিএনএ কিন্তু প্রোটিন তৈরি করে না। ডিএনএ হচ্ছে ইনফরমেশন। প্রোটিন তৈরির জন্য যেই ইনফরমেশনটা লাগবে সেটা হচ্ছে ডিএনএ তে থাকে। সো ডিএনএ ইজ দ্য ইনফরমেশন, and by that information, following using that information, ribosome can produce the DNA protein. Okay, now for every protein there is a different portion in our DNA. So for example, insulin is a protein. Okay, So for insulin, there is a part, dedicated part in our DNA. I mean, okay. So that part contain all the information by which the insulin can be produced. Okay. So that part is called gene. So sometimes that's why sometimes we call insulin gene. Okay. Usually we call the both the protein and the gene same way. Insulin protein, insulin gene. Okay. So insulin gene is that part of our DNA which contain the information necessary to make or produce the insulin protein. Okay, so this is gene actually. So for every protein there is a dedicated DNA portion, DNA portion, and that each portion is called gene, GENE. Okay, and we know that actually the gene we are and every gene have two copy in our body. One we get from our mother, another we get from our father. Okay. That's why each of us are different. But you guys now are clear about what is gene? বুঝছো তো জিন কি জিনিস?
জি
Yes,
স্যার।
sir.
Okay. So that's why we say gene encode the protein. I frequently, I frequently say this word encode. Gene encode for the protein. Okay. Then what is genome? Now sometimes you will heard that the genome sequence. Genome means the all the gene, the total DNA is called genome. So when we call gene refer to one that is small part which encode for one protein. But when we say genome refer larger part. So for example, if we say human genome means the total DNA of a human genome. Okay. Basically, that's why when we sequence the whole, all the nucleic acid, we don't say gene sequence. We call genome sequence. Basically, the method is same, right? Gene and genome. What is the main difference? Gene is a small part which is encoded for all one protein, and genome is the total part. আচ্ছা এগুলো তোমরা শিখবা আস্তে আস্তে, তোমাদের জেনেটিক ইঞ্জিনিয়ারিং কোর্স আছে। আচ্ছা কারো কিন্তু যে কুইজের স্ক্রিপ্ট নিয়ে কোশ্চেন থাকলে করতে পারো, আদারওয়াইজ আমরা কিছুক্ষণ পড়াশোনা করি, অল্প কিছুক্ষণ। I ask you to study which lecture 11 or 12? এই লেকচার 11 কিভাবে আলোচনা করা হয়েছিল? আমি কিন্তু ভুলে টুলে যাই, পরে কিন্তু শুধু শুধু আমাদের সময় নষ্ট হবে। Yeah, the lecture 11 was done, right? Okay. আরে তোমরা লেকচার 12 পড়ছো? Have you studied lecture 12? এই না পড়লে বকা দিব না, হ্যাঁ বলো অসুবিধা নেই যে না স্যার পড়ি নেই।
না স্যার পড়িনি স্যার।
পড়া হয় নাই স্যার।
Okay, thank you.
স্যার পড়া হয় নাই।
Okay, it's good to be honest, right? পড়ো নাই সেটা তো আমি বুঝছি, হ্যাঁ। আচ্ছা একটু পড়াশোনা করো। Okay, I'll give you a brief about lecture 12, but if you have already studied lecture 12, you can ask me question. Okay. So in lecture 11, we have already learned about the characteristics and structure of virus. So this lecture 12, is about the replication of virus. Okay. How the virus increase their number. How the virus replicate, how they increase their number. So in this chapter, okay, there are different viruses, means like a plant virus, animal virus, bacterial virus, which we call bacteriophage. Okay, so I mean, as example, to understand the replication, as example, we will talk about or discuss about the bacteriophage. Okay, the virus which infect bacteria. Okay. Okay, so when the virus replicate, so the bacteria, I mean, also some animal virus do that. I mean, when the virus replicate, they can follow two different pathway. Okay. Okay, just remember now they can follow two different pathways. So what pathway? To understand that, I'll directly move to this slide.
আচ্ছা স্যার একটু ইন্টারাপ্ট করলাম
Yeah, sure.
সো আপনি আমাদেরকে একটু প্রি অ্যাডভাইজিং নিয়ে কিছু বলতে পারবেন লাইক হোয়াট সাবজেক্টস হোয়াট কোর্সেস উই শুড টেক ফর দ্য নেক্সট সেমিস্টার বা কি
Okay. Pre-advising. Good question. ভুলে গেছিলাম। দাঁড়াও। Course. We should talk about little bit pre-advising. তোমাদের, I mean, when you guys are pre-advising?
19th
19th.
yes
Hey, okay, there is no microbiology senior here. Okay, it would be better. Is there any microbiology senior here present in the class right now? No? Okay. আমি তোমাদের সিনিয়ররা থাকে মানে ফিজিক্যালি খোলা থাকলে না সিনিয়রদের কাছ থেকে এসব পেয়ে যেতে আর কি। I mean, the seniors, they know much better than me about the taking the course. Okay, anyway, so okay guys, so you know what kind of courses you should take. Like there are different type of courses: core course, elective course, and we call COD and GNET course. So GNET course is right, we call them general education course. And I forget, I mean, previously you guys should take 42 credit from the general, I mean, including GNET, general education, and course, a COD course, outside department course. Okay. In general, sometimes we just call it COD or GNET. Previously, it was 42 credits, but I think for you, you have to take 45 credits. Okay. And among the 45 credits, you have some option from where you have to take what? 21 plus 3. 24 credit, and from some, and there is another list from where you have to take 21 credit. So in total 24, 21, 25. Okay. So in every semester, you guys can choose some like GNET or COD course and some core course. এটুক তো বুঝছো নাকি? Okay, have I ever shared the degree plan with you? I mean, a tentative degree plan by which you can guess what kind of courses you should take.
নো স্যার
তোমাদের সাথে এরকম কোনো ডিগ্রি প্ল্যান কি আমি শেয়ার করছিলাম?
না স্যার
ওকে, just give me 1 minute please, just 1 minute. এখানে ফাইল এড করে কেমনে? এক মিনিট। Okay, I have uploaded a degree plan in the classroom. Okay, it's a tentative degree plan, so don't consider it as a fixed one. You have your liberty to choose courses, okay, according to your conveniences. So it's just a tentative plan. So can I share this plan during this class? Okay, just 1 minute. Mm, যে তোমাদের সিনিয়রদের সাথে তোমাদের কোনো কন্টাক্ট হয় নাকি?
স্যার আমাদের একটা ফিয়ার গ্রুপ আছে, আর আমাদের মেন্টর আছে নাবিলা আপু।
তোমাদের মেন্টরের নাম কি?
নাবিলা খান।
নাবিলা নাবিলা খান?
জি স্যার।
Your mentor is Nabila Khan, right?
জি স্যার।
Okay, now Nabila Khan is a very nice person, right?
Yes sir.
So feel free to ask her anything. Don't worry, huh?
না স্যার, আমার কোশ্চেন হচ্ছে আপনি কি কোনো কোর্স নিচ্ছেন যেটা আমরা নিতে পারবো নেক্সট সেমিস্টার, লাইক মাইক্রোবায়ো 1 or 2 বা ল্যাব কোর্সটা?
Actually, no. I mean, I take a 200 level course. I mean, at 100 level, I take only Mic 101. In 200 level, I take Mic, no, not Mic, BCS 202. BCS 202 is an elective course for you. Okay. And BCS 202 is like a common course for both microbiology and biotechnology program. For biotechnology, it's a compulsory course, but for microbiology students, it's an elective course. BCS 202. And then what course I take? In 300 level, 309, immunology and vaccine development. 400 level, 407. 407 is public health. It's related with public health. Epidemiology, actually. 407, epidemiology, and 403, quality control of food, fish, and beverage. That's the courses I usually take. Okay, so you guys know what you have to take in the next semester, right? So, for example, you can take HAM,
স্যার
Web.
এটা তো আরএস মনে হয়,
আচ্ছা, তোমাদেরকে আরএস এর কোর্স দিচ্ছে না এবার? I mean,
স্যার আরএস আমাদের থার্ড সেমিস্টার।
RS your third semester. Okay, in RS you will take these 2 semesters. Then in that case you can take ENG 102. So I think all of you didn't get the ENG 101 this current semester, right? Freshers?
না স্যার, আমার ইংলিশ 102 ছিল।
Or you have English 102. So I think some of you they didn't get the English 101. So you have to take English 101 must. Okay, you have to take the English 101. So these courses like Mic 101 you are doing. So BCS 101. বিসিএস 101 আমি জানি না সবাই বিসিএস 101 এবার করতেছে কিনা, যারা করতেছে না তারা আর কি যে ইংলিশ 101, বিসিএস 101, যারা I mean those who already doing this in this semester you can take my English 102. These are GNet course. Okay, so bio 101 is not mandatory. Okay, it's a what can I say, it's a useless course. I mean it's not for you guys actually. I mean it belong to the GNet course, so if you want you can take it. Okay, it will fulfill your purpose. You must have to take 45 credits at a GNet course, so it will fulfill that purpose. But it's a boring. I mean, it's bio 101. You are biology student. You already know these things. So if you want, you can take it, or otherwise, alternatively, you can take like physics or chemistry. Okay, so it's up to you. Whatever you like. Then English 102. Take Mic 102, BCS 201, CSC 101, this kind of things. ঠিক আছে, বোঝা গেল ব্যাপারটা?
জি স্যার, একটু
And also feel free to ask Nabila. I think she can give you better suggestion regarding these things. কোর্স নেওয়ার সময় একটু সিনিয়রদের সাথে আলাপ করে নিও। I mean, the private university system is not like the public university system. Like say for example, in my university there was options like there was core course and elective course. But the thing, I mean, the core course are mandatory, and the things of elective course was like that. So you have to take two elective course, and there was only two options. Okay. তা আমাদের ইলেকটিভ কোর্সটা এরকমই ছিল যে দুইটা ইলেকটিভ কোর্স নিতে হবে, দুইটাই অপশন। তো তোমাদের মানে তোমাদের ব্যাপারটা এরকম না। Okay, you guys have several options, so you can take, I mean, the core courses are mandatory. You must have to take the core courses. But for the other courses like the elective and Gen Ed or COD courses, course outside department courses, you have several options. So you can choose which one you like. Okay, আমরা পড়াশোনায় ফেরত যাই। এই কোয়েশ্চেন থাকলে করতে পারো কিন্তু, হ্যাঁ, কোনো সমস্যা নেই। Presentation শুরু হয়েছে, হ্যাঁ হয়েছে। Okay, so where we were, viral replication. Okay, so as I said, the virus, they can follow two cycle in their replica. I mean, they can follow two pathway during their replication. Okay, one is called lytic, another is called lysogenic. Okay, so what are the differences between these two cycle, and why one is called lytic, why another is called lysogenic? To understand that, I directly move to slide number 7. Okay, so in this slide you will find the both lytic and lysogenic. Okay, so why they are shown together? Okay, so basically, you know, the same virus, it can follow both. It can follow either lytic or lysogenic. Okay, so here as an example, the life cycle of a bacteriophage which name is bacteriophage lambda has been discussed. Okay, bacteriophage lambda as an example. Okay, usually the basic is same. I mean, the basic steps of lytic and lysogenic cycles are same for both, like all the viruses. The only difference are usually in the first phase. I mean, how the virus enter into the host cell. লাইটিক সাইকেল এবং লাইসোজেনিক সাইকেলের যে স্টেপস থাকে, স্টেপস গুলো বেসিক্যালি সেম আর কি। হ্যাঁ, ওই যে ওই ব্যাকটেরিয়াল ভাইরাসের ক্ষেত্রে, মানে যারা ভাইরাসকে ইনফেক্ট করতে, ব্যাকটেরিয়াকে ইনফেক্ট করতেছে, তাদেরটা অ্যানিমাল ভাইরাস ইজ সেম। ওকে, সো ডিফারেন্সটা হয় শুরুর দিকে, মানে ভাইরাসটা যখন সেলের মধ্যে এন্টার করে আর কি। ওকে, it's a slight difference. Okay, anyway, so let's try to get idea about the life cycle of this virus as an example. Okay, lytic cycle and lysogenic cycle. So this is a bacteriophage, that means it will infect bacteria, so its host is bacteria. So what happened at the first step? Okay, at the beginning, this virus, they attach with the bacterial cell. This is the bacterial cell and this is the phage. Okay, it has attached with the bacterial cell. That means this bacterial cell is the host for this virus. So after attachment, all the body part of the virus remain outside. Only the nucleic acid get inside the cell. So you can see here. Okay, so first the virus attached with the host cell and they inject their nucleic acid inside the host cell. Okay, only the nucleic acid. All the other part means basically they have the protein code. Okay, if they have envelope, then the envelope. So the protein code and the envelope, I mean, if they have envelope, they remain outside, and only the nucleic acid get injected inside the host cell. Okay. Okay, so for this injection, actually, this virus they have to make hole, or they have to puncture the bacterial cell wall. Okay, some virus they have their own enzyme to make hole or puncture into the host cell wall. Okay, okay. So anyway, they attach with the, the virus attach with the host cell. They make hole or puncture the bacterial cell wall, then inject its nucleic acid. Okay, so now once the viral nucleic acid is inside the host cell, they become circular. Okay, so usually the viral host nucleic acid, they're not always circular. Okay, sometimes they may be filamentous. Whatever they are, once they are inside, at least when the virus they inject their nucleic acid, they inject it as in a linear form. এরকম সুতার মতো linear form এ পাঠায় আর কি. So once the viral DNA, once the viral nucleic acid is inside the host cell, they become circular like this. এই কেউ যদি কোনো কিছু না বোঝো, জাস্ট ফিল আমাকে জিজ্ঞেস করো, হ্যাঁ? If you don't ask anything, I mean my word or my sentence, whatever, just feel free to ask. Okay, they become circular. Okay, so now these two, until this, it is same for both lytic and lysogenic cycle. So the difference start from now. So after this, now this virus has two options. Either it can start the lysogenic pathway or cycle, or the lytic cycle. Okay, so how there is some controlling system. Okay, I will talk about the controlling system. I mean, how the virus decide, or what determine whether the virus will go to lytic or lysogenic. There is a system. মানে একটা সিস্টেম আছে, যে কন্ট্রোলিং সিস্টেম আছে, কিছু ডিটারমাইনিং ফ্যাক্টরস আছে যারা ডিটারমাইন করে যে এরপরে ভাইরাসটা লাইটিকে যাবে না লাইসোজেনিকে যাবে। ওটা আমি একটু পরে আসতেছি। Okay, so now say for example the virus enter into the lytic cycle. So what happened, and after this step, the viral nucleic acid, they use the host machineries. Machineries means what? The host machineries like ribosome, the host protein synthesis, and nucleic acid replication system. Like for protein synthesis, they use the host ribosomes. For making copy of the nucleic acid, the viral nucleic acid, they may use the polymerase. Okay, for making copy of the nucleic acid, the virus need enzyme. Okay, polymerase enzyme. Okay, so they say DNA polymerase enzyme. For making copy of DNA, they need DNA polymerase enzyme. So they use the host enzymes, ribosomes. Okay, sometimes the virus they may have their own enzyme, but they never have their own ribosome. Okay, so they use all the host machineries, I mean the virus, and by using these host machineries, they synthesis part of their body. Like they say, what are the part of their body? They synthesis, they make copy of their nucleic acid. They synthesis the capsid, right? The protein required to make their capsid, right? Or the protein outer layer. Okay. So basically, using the host machinery, the virus produce all those structural unit of their body, and then those parts get assembled and form the full viral particle or complete viral particle. Then the host cell, it gets destroyed, which we call lysed. Okay, lysis means breaking apart. Okay, lysis means just tearing it down. Okay, so okay, anyway, okay, so the host cell get lysed, and this virus they get released outside the cell, and now they can infect another cell. Okay, so as the host cell getting lysed, it is called lytic cycle. Okay, because in this cycle the host cell is simply get lysed or destroyed. Okay. Okay. So now, what happened if the virus enter into the lysogenic cycle? So now, say for example, the virus. I mean, after this, I mean, so as these are shown separately, you can see that these two steps, this step and this step, these are same, right? So after this step, it can enter the lytic cycle or the lysogenic cycle. So now we have talked about the lytic cycle. So what happened in lysogenic cycle? So now the virus, it say this virus has enter into the lysogenic cycle. So what happened if it enter into the lysogenic cycle? So this viral DNA, this circular, viral nucleic acid, or actually it will be DNA. Okay, let me add one thing here. There are RNA virus or DNA virus. So if it is a DNA virus, it is already DNA. Okay, so if it is a RNA virus, there will be an additional step. RNA virus হলে একটা additional step থাকবে, কি step? That the RNA will become DNA. Okay, so that's the only difference. Otherwise, it will be same. I mean, for the RNA virus, they need to we call it reverse transcription. Whatever, first the RNA need to changed into DNA. Okay. Otherwise, that will be the only additional step for RNA virus. So anyway, so this is DNA. So if it enter into the lysogenic cycle, this DNA, it will get integrated into the host DNA. Integrated. So if from the figure you can see, right? It has integrated into the host DNA. Okay. So the process by which this integration can occur is called recombination. Okay. So right now, just remember this word, recombination. Recombination is the molecular process by which these things can happen. Okay. Okay. So this integrated viral DNA, it is called prophase. It's just the name. Okay. So prophase means the integrated. The prophase referred to the viral DNA which is integrated into the host DNA. Okay. Okay, so once they're integrated into the host DNA, the viral DNA, the prophase integrated into the host DNA, they can remain in this state for a long time. Long time means days, months, year, centuries. Okay, they can remain integrated for years after years, for months after months. Okay, so what happened during this time? So as they are part of the host cell now, host DNA. So whenever the host cell replicate, the number of the host cell increase, automatically the number of the prophase increase, right? Whenever the host cell make another copy, they also make a copy of the DNA. Okay, and as the viral DNA is integrated into the host DNA, so whenever the host make a copy of its DNA, the viral DNA automatically get replicated, right? ভাইরাসের ডিএনএটা তো হোস্টের ডিএনএর মধ্যে ইন্টিগ্রেট অবস্থায় আছে। সো যখনই হোস্টের ডিএনএ রেপ্লিকেট করে, হোস্ট সেল নিজে রেপ্লিকেট করে, নিজের সংখ্যা বাড়ায়, তখন তার তো কি ডিএনএটা হচ্ছে একটা কপি বানাতে হয়। সো কপি বানানোর সময় এই ভাইরাসের ডিএনএ অটোমেটিকলি কপি হয়ে যায়, কারণ সে তো এখন একটা পার্ট হোস্টের ডিএনএ। হ্যাঁ, তার তো আলাদা ইন্ডিভিজুয়াল কোনো এন্টিটি নেই, পরিচয় নেই। ওকে, সো এনিওয়ে, সো বাই দিস ওয়ে the number of the prophase it get increased. Okay, so so now as I said, they can be like this in the this is called the lysogenic form. Okay, lysogenic form. So they can be, the viral DNA can be integrated in the host for months after months, years after years. Okay. So now if the virus want, they actually can get disintegrated. They are now integrated with the viral DNA, host DNA. So the virus can detach from the host DNA and get separated. Okay, well, it don't happen without any cause. There are some factors which influence this detachment. Okay, there are some factors. I'll talk about this later in the next slide. So now, say for example, the viral DNA is integrated in the host DNA. So there are some factors which can induce that the viral DNA will, which can induce the detachment of viral DNA from the host DNA. So at the time, when the virus need to separate it from the host, the viral DNA, they disintegrate it from the host DNA and become separated again, like this way. Okay, so this is the cycle. Then they enter into the lytic cycle and get released. Okay, so this is called lysogenic cycle. So I guess you got the idea of what lytic cycle and lysogenic cycle is. The same virus, as I said, the same virus can follow both. So the same virus, after infection, the virus, they can directly get into the lytic cycle, okay, or they can get into the lysogenic cycle and remain in the lysogenic cycle for several times, and then they can again re-enter into the lytic cycle. Okay, all are possible. So are you clear until this one?
Yes, sir.
এই পর্যন্ত তো
Yes,
ক্লিয়ার
sir.
আছে নাকি? আর ক্লিয়ার না থাকলে অসুবিধা নেই, পড়লে অনেক ধরনের কোশ্চেন থাকবে, তখন আমরা জিজ্ঞেস করি আর কি। Okay, so right now, in this photo, actually, I briefly described the lytic cycle. That lytic cycle itself has some steps. Okay, so you will find the steps here: attachment, penetration, early steps, replication, biosynthesis, maturation, release. Okay, so this photo, it depicts the detailed steps of the lytic cycle. Just try to read. Okay, if you don't understand, please ask me question in the next class. Okay, these are easy thing. I have already actually discussed this thing, so একটু পড়ো ভাই, না পড়লে তো কোশ্চেন জাগবে না আমার। Okay, so okay, so okay, this I already discussed. Yeah, the steps of, these are the steps of lysogenic cycle. Okay, if you read, you'll understand easily. Okay, then sorry, I told you one thing, that the same virus, it can decide in which cycle it will enter, lytic or lysogenic. Well, actually, there are some factors which determines in which cycle the virus will enter, the virus will follow or enter after this state. Okay, so what are those factors? What are those determining things? Well, there are some proteins. Okay, everything in the living world, inside the living thing's body occurs is maintained or regulated by protein. Okay, everything, like say inside our body. So we are human, in our body everything is controlled by different proteins. Okay, সব নাটের গুরু হচ্ছে প্রোটিন, হ্যাঁ, তারাই সবকিছু করে সব সেলের জন্য। It can be a bacteria, it can be human, it can be whatever. Okay, everything is controlled by different proteins. Also here there are some proteins which determine whether the virus will enter the lytic or lysogenic cycle. So what are those proteins? Okay, what are those proteins? There are different proteins. Okay, so one is repressor protein. There is one protein which is called repressor protein. There is another protein which is called viral activator protein. Okay, say okay, so for example, okay, now in every step of this like both cycles, say for example the lytic cycle, okay, the viral DNA, the viral DNA, okay, it is here and it has to use the host machineries to synthesis all of its body parts. Okay, so for this work the virus needs several proteins. Okay, in these steps there are several proteins which are involved in doing this kind of works. Okay, so now once the viral DNA is inside the host cell, they try to they begin to synthesize different proteins. তারা বিভিন্ন ধরনের প্রোটিন সিনথেসিস শুরু করে আর কি, হ্যাঁ, হোস্ট মেশিনারিজ ইউজ করতেছে হোয়াটএভার, প্রোটিন সিনথেসিস শুরু হয়। সো নাও দ্য থিং ইজ হুইচ ওয়ান ইজ দ্য ফার্স্ট প্রোটিন being synthesized, মানে সবার প্রথমে কোন প্রোটিনটা তৈরি হবে, this is important. Okay, so which one is the first protein? So for example, if the repressor protein is synthesized first, repressors protein, there is a protein which is called repressor protein. Okay, so if this protein is synthesized first, I mean once the virus is inside the host and the repressor protein is synthesized first, so what happened, this protein stop or the inhibit the synthesis of all the other proteins which are necessary for the lytic cycle. So if repressor proteins is synthesized first, synthesis of all the other proteins necessary for lytic cycle, they are stopped. So the virus enter into the lysogenic cycle. Okay. Now, what happened if not the repressor protein, but the viral activator protein C2? Okay, it's the name of this protein. The viral activator protein C2 is synthesized first. So if this protein is synthesized first, then the virus will not enter into the lysogenic cycle. Rather, actually this protein, it suppress the synthesis of this protein, repressor protein, but it allow or induce the synthesis of all the other proteins necessary for lytic cycle. So if this protein is synthesized first, then the virus will enter into the lytic cycle. Okay, so that's it. These are the determining factors of the proteins which determines whether the virus will enter into the lytic cycle or lysogenic cycle. Okay, so now say for example the virus is in what, the virus is in the lysogenic form. Okay, its DNA, it is, the virus is in prophase, that is, it is integrated in the host cell, host DNA. So now what determine that they will, I mean the virus will get detached from the host cell? There are several factors. So for example, there are some chemical factors, light, there are some light factors, maybe say laser light or some UV type light. Okay, I think there is something here where it is okay, sorry, I forget the name, which can I mean the which factors can I mean induce the separation of prophase from the host DNA. But there are some several factors, like there are some chemicals or like light. Okay, like ultraviolet type lights, not very powerful UV light. A powerful UV light will destroy the DNA. Okay, so by those kind of inducers or factors, they can I mean, so the factors which control that with the excision or separation of prophase from the host DNA. So say for example light is a factor. So if the light means, say for example UV light, okay, so certain wavelength UV light is a factor to induce the separation. Then what will happen when those factors, I mean this bacteria, I mean which has the integrated virus, is exposed to that factor, then the synthesis of repressor protein will get down. Okay, and the synthesis of this protein will be activated, and then again once this synthesis of this protein is activated, the other proteins which are necessary for lytic cycle which begin to being synthesized, and then the virus will enter into the lytic cycle from this lysogenic. Okay. Okay. Okay, that's all for today's class. If you have any question, you can ask. Or okay, next class we have a quiz. So after the quiz, please study and ask me question in the next class. We have to understand virus clearly, right?
Yes, sir.
Okay. What is this release of? Okay, then I will talk about this in the next class. Any question? Sir, anyone have any question?
No, sir.
Okay. Then best of luck. Okay.
Thank you, sir.
Okay.
Thank you, sir.
The next class we have a quiz. It will start from 11. Okay. So if you have any problem, just feel free to knock me in the. I mean, you can directly call in my cell phone or WhatsApp. Either is okay.
And sir, assignment কত mark carry করবে?
বুঝি না।
Assignment কত mark carry করবে?
Oh, assignment will be 20. Okay.